So hello everyone. Welcome to the RISE ECE and Neuroengineering Distinguished Lecture Series. This is the last of our summer series. We will remain virtual in the fall. So if you would like to hear about additional seminars like this and more, please connect with us through our website, neuroengineering.rice.edu, on Twitter @RiceNeuro, or Facebook RISE Neuroengineering Initiative. Today, it's my distinct pleasure to welcome back RISE alumnus Dr. Lihong Wang as our distinguished speaker. Lihong Wang, Professor Wang earned his PhD degree here at RISE under the supervising of a Nobel Prize laureates, Dr. Robert Kroll and Richard Smalley, as well as Dr. Frank Tichtel. Professor Wang is currently the Bren Professor of Mechanical Engineering and Electrical Engineering at the Andrew and Becky Chang Department of Mechanical Engineering at Caltech. And before he joined Caltech, he was formerly the Jean Baer Distinguished Professor of Biomedical Engineering at Washington University in St. Louis. Professor Wang is renowned for his contribution to the field of photoacoustic imaging technologies and invented the world's fastest camera with more than 10 trillion frames per second. He has made numerous contributions in bioimaging and has won many awards for his original contributions. For the time's sake, I would only name a small fraction. He received the NSF Career Award, the NIH First Award, NIH Director's Pioneer Award, NIH Director's Transformative Research Award, the NIH NCI Outstanding Investigator Award. He also received the IEEE Technical Achievement Award, the IEEE Biomedical Engineering Award, SPIE Brenton Chan's Biomedical Optics Award, IPPA Senior Prize, and OSA Michael S. Feld Biophotonics Award for seminal contributions to photoacoustic, tomography, and light speed imaging. He is a fellow of the AAAS, AIMBE, Electromagnetics Academy, IAMBE, IEEE, OSA, and SPIE, as well as a foreign fellow of CLS. He received an honorary doctorate from Lund University, Sweden. And in 2018, he was inducted into the National Academy of Engineering. With that, please join me in welcoming Professor Wang back to RISE.
Thank you, Dr. Wang, for the very kind introduction. It's a tremendous honor to present at one's alma mater. So thank you all for joining me as well in my presentation. I'll be talking about two research directions my lab has been taking on, photoacoustic tomography and compressed ultrafast photography. The first one targets deep penetration, especially in biological tissue, scatter media in general. And the second one targets imaging speed. The very first question is why optics? For medical imaging, we have a lot of choices, right? You walk into a hospital, you got MRI, X-ray, CT, ultrasound, PET, and even SPECT. And now optics is very special. If you start from the physics perspective, we know light-matter interaction occurs at a molecular level. So there are fundamental roles molecules are playing in biomedicine. We really cannot ignore this direction. We want to have molecular specificity. By detecting molecules, we provide in vivo functional imaging similar to functional MRI, in vivo metabolic imaging analogous to PET, positron emission tomography, in vivo molecular imaging of gene expressions or disease markers, even in vivo label-free histologic imaging of cancer or any other diseases without tissue excision. And so there's a host of imaging capabilities that optical imaging can provide, but we face daunting challenges. Now, when you look around the world, turbidity is almost ubiquitous. All the materials you see, with a few exceptions, are turbid or scattering. And you look around, you see trees and snow, and they're all turbid. Now, water is clear to some extent. You look at our body, biological tissues, nearly all tissues are scattering, with the exception of our eye. And so we have to deal with turbidity. The other challenge we face is speed. Light travels at a terminal at ultimate speed. Nothing can propagate faster than the speed of light. And so optical phenomena are simply too fast for modern imaging devices to capture. And so if you look at sonic events, like this supersonic Mach cone, and this is captured by NASA, right? So light travels 5 orders of magnitude faster than sound. So to capture a sonic event, and we can use light. And this is, you can see this cone structure, and because this plane is traveling at a supersonic speed. Now there's an optical counterpart. And so if you have a light source propagating at a superluminal speed, meaning the speed of the source is greater than the speed of propagation of the light wave in the material, then you can generate a counterpart of the sonic boom, but we can never capture that cone structure because none of our devices are fast enough to capture that event. In fact, you might remember your college of physics where we talked about Cherenkov radiation. It's exactly due to the superluminal radiation. So let's come back to the first problem. If you look at light scattering, this is a piece of tissue. We're only talking about the first 2 millimeters beyond the skin surface. And you can see light propagation starts to diffuse very quickly. Within about a millimeter, the photon propagation transitions from the early ballistic regime to the later diffusive regime. And this presents a huge challenge for high resolution imaging at depth. Starting roughly 350 years ago, our field has invented optical microscopy. You can see subcellular structures, you get fantastic spatial resolution, but the penetration is limited to about 100 microns. This is something we call the optical aberration limit. And that's basically 1 entry path in scattered biological tissue. Now, we were basically stuck at that limit for hundreds of years until laser was invented. By using lasers' special properties, we can figure out new techniques such as confocal microscopy, 2-photon microscopy, or optical coherence tomography. Now, all of a sudden, we improve the penetration by order of magnitude. And now we face the next challenge, which is about 1 millimeter in scattered biological tissue, which is called the optical diffusion limit. We stagnated at that limit for a long time until photoacoustics was invented. And so now, instead of using just the ballistic or quasi-ballistic photons, we can actually tolerate scattering. We allow photons to scatter multiple times. Now we need to find a way to retrieve the spatial information because after photons have gone through random walks, it's really hard to retrieve spatial information directly from the photons. And we have to think of a different way to retrieve the spatial information. And this is why we introduce acoustics into the picture for a hybrid model. And that allows us to get optical contrast and ultrasonic resolution in a single modality for very deep tissue penetration relative to standard optical imaging. And so now we're talking about not just millimeter scale penetration, but we're really talking about multiple millimeter all the way to multiple centimeters of penetration for whole body small animal imaging, for whole organ human level imaging. Now we do face the next challenge, which is something I call the dissipation limit, because the energy density gets quite low after a certain depth. And there's an active field called wavefront engineering or time reversal. And especially if we can introduce a virtual guide star, we might be able to time reverse light to generate hotspots. And using the hotspots, we can generate imaging information as well. But this field is not mature yet for practical applications. So I'll focus on photoacoustics. In fact, Alexander Graham Bell first discovered photoacoustics in 1880. And so there was no laser, and he used the sunlight. Yeah, he encoded sound wave into sunlight, propagate sunlight, and then on the other side, you can convert sunlight. These are modulated version of the sunlight because the intensity is modulated by sound, and you can actually hear directly without the standard demodulation, and because that modulation is at audible frequency. And so, of course, this is just the physical concept at the time, and as a communication device, this never took off because it requires line of sight propagation. And now, can we introduce a set of modern concepts, laser, ultrasonics, tomography, and computer, and combine with this very old physical concept, photoacoustics, to produce a new imaging technology called photoacoustic tomography. In photoacoustic computed tomography, we normally start with a nanosecond laser pulse. We broaden the laser beam, such as the energy per area is within the safety limit. So the ANSI regulates energy per area. We call that exposure, or sometimes called fluence. And so as long as your energy per area is within the limit, you're very safe to use laser light. And we allow photons to scatter multiple times so we can provide deep penetration. When photons are absorbed, we generate rapid heating. Every millidegree gives you a detectable pressure. So 1 millidegree gives us roughly 800 pascals of pressure, which is detectable with ultrasonic transducer already. But within the safety limit, typically we can generate hundreds of milledegrees, and you're still safe. And by your SNR, signal-to-noise ratio, is very high for us to produce a high-quality image. So unlike standard optical imaging, we don't detect return photons. We detect the returned ultrasonic wave instead. Now, if you look at acoustic scattering coefficient, it's orders of magnitude lower than the optical scattering coefficient. So by detecting the ultrasonic wave, we can form a very sharp image, and that's quite deep relative to the standard high-resolution optical imaging. And so using this combination of light and sound, we're combining optical contrast and ultrasonic resolution in a single modality. Let me start with a very simple analogy on how we can form an image. Let's say we have a single point source like a thunderbolt. And when you see the lightning, you can reset your stopwatch to time 0. When you hear the thunder, you can record a time delay T1. Now, of course, if you have 3 such detections, and then you can triangulate the point source, you can figure out exactly where that point source is. So photoacoustics is somewhat similar to that, except we don't have a single point. We have a volumetric source that's heterogeneous, because that's the exact nature we want to figure out. Because if you can figure out the distribution, the volumetric distribution of the sound source is induced by laser absorption, laser light absorption, you generate a 3D image. And that's the goal we have in mind. So 3 detectors will not be enough. In fact, to form a 2D cross-sectional image, we need hundreds of detectors. And so this is the initial version of the inverse spherical radon transform. And so if you have a 3D unknown object, then we essentially bathe the object with photons, broad beam illumination, and you generate an instantaneous volumetric source because the light speed is so high, we can ignore the propagation time of light. And you have a delta T source. And now you have a number of ultrasound transducers outside, and for a given detector on the detection surface, at time T, we're essentially integrating on this spherical surface. Right? This spherical surface integration is called a spherical radon transform. Now, radon never inverted this problem, so our field had to work on this problem really hard. In the early days, we had one solution for each geometry. It was kind of awkward to use, and sometimes we had to use infinite series to come up with a solution. And so it turned out we have a closed form solution, and that's universal to all the 3 detection geometries that are standard, namely planar, cylindrical, and spherical. And so P0 is the pressure, we call that initial pressure induced by the laser pulse. That's due to the light absorption. And at first glance, that's a mechanical parameter, but this is proportional to the optical index deposition, and namely proportional to the optical absorption coefficient. And so we can actually retrieve optical contrast from P0. And so what we detect is P at detector position R0 at time T. So it's a time sequence. What we do is we use solid angle to unify the different detection geometries. And you have to account for time delay, because when the signal is received, there's a time delay, and we'll back project, and we have to time reverse that back into the object. Now, if we use the pressure only, it is very similar to the ultrasound beamforming, which has been used in radar, in sonar, right, in ultrasound imaging as well. Now, if we introduce this high frequency or temporal derivative component, and we can actually get an exact solution. So mathematically, this is very exciting to get at a time. And in fact, this is still the most widely used algorithm. We call this universal back projection algorithm in our field. In 2003, we achieved this first set of functional photoacoustic images, also the first set of in vivo photoacoustic images in small animals with intact scalp and skull. And by wiggling one side of the whiskers, the contralateral side of the brain was activated, and we can see the hemodynamic change due to the whisker stimulation on one side, on either side. And so this was extremely exciting at the time. In fact, it kicked off exponential growth of our field, as you can see right here. After 2003, and our field has experienced exponential growth with a time constant of roughly 3 to 4 years. Since 2010, the conference on photoacoustics has become the largest at the 20,000 attendee Photonics West, and now is the largest by far. Since last year, the field has published each year over 1,000 peer-reviewed journal papers and 28,000 citations within a year. And so this is a list of some of the top most cited papers. So we've been very fortunate to earn the top 2 cited papers. And one was the demonstration in 2003. The first is functionally vivo imaging. And the second one, 3 years later, was the first 2D photoacoustic microscopy image. And so this paper really continued our exponential growth and played an important role in our field. And by now, there are at least 19 companies that are open. They've been commercialized in photoacoustic tomography and microscopy. And I know there are also companies who prefer to be anonymous. They're also commercializing photoacoustic technologies. So this is an extremely active field. And there are commercial products out there for small animal imaging, and companies are going through regulatory approval for clinical imaging as well. So for the 2003 work, we demonstrated the principle essentially using a single element ultrasound transducer, and that took about 20 minutes to get a 2D cross-sectional image. And that's obviously too slow for practical applications. And so finally, after years of hard work, we implemented a single shot version. We call it single impulse panoramic photoacoustic CT. And so with a single laser pulse, we get a 2D cross-sectional image. And so that's extremely fast. Data acquisition is done within tens of microseconds, limited by the acoustic transit time across the field of view. And so we can configure the system for trunk imaging. And you start with the right laser, and you send the laser beam through a conical lens to produce a hollow cone beam. We generate photoacoustic wave. And so you want to conserve every bit of laser energy. And that's where for trunk imaging, the hollow cone beam is the optimal configuration. And we use a full ring ultrasonic transducer. And although I'm showing only half of the ring here, and this full ring allows us to view the object from all possible angles. That gives us best image quality. Of course, Nyquist, whenever it's possible, and we should conform to the Nyquist sampling criterion. And so this is a spatial Nyquist, right? So you want to sample at least 2 points per acoustic wavelength. And so 512 turned out to be the right number. And so we can sample with minimum streaking artifacts. Preamplification is very important because the signal, especially if you want to image deep, the signal is very weak, and you want to conserve every bit of signal to noise ratio. And so you want to amplify your signal as soon as you get the electronic signal, instead of waiting for cable noise to creep in, and then your SNR is reduced. It's very hard to retrieve the lost signal to noise ratio. We use a one-to-one data acquisition, and so with a single laser shot, we can capture the image again within tens of microseconds. Then we can repeat our laser pulse to get the next 2D image. Of course, we use the computer to run the inverse spherical radon transform, which is the inverse problem for image reconstruction and for image display as well. Here's a close-up of the key components, and you have a cone beam, hollow cone beam, and we use ultrasonic focus in the elevational direction to generate spatial resolution directly, but your XY or horizontal plane resolution comes from the inverse solution. The system could be reconfigured for brain imaging. And so here, for brain imaging, a solid cone beam is an optimal design, optical illumination geometry to conserve the laser energy. And so we use a diffuser only. These are usually engineered diffusers, so you optimally conserve all the energy instead of a standard diffuser. And we get a solid cone beam on the head of the small animal to be imaged. And just to give you a few examples, and this is a whole body, whole trunk imaging, and the red line shows the current plane being imaged. Each shot is acquired within 20 microseconds, and then we repeat our laser pulse at 50 hertz. And so we get 50 frames per second, and 50 hertz is the current limit of the laser rep rate. And if you repeat your laser pulses, you can even achieve a higher frame rate. And you can see different structures. And this is close-up of this red box. You look at the intestine, and you can look at even the heartbeat. You can see a torsion motion actually of the heart, the lung structure, the liver, vena cava, portal vein, spleen, and kidney structures. You can see some of the symmetries on two sides of the kidneys. So let me give you some detailed structures. For this, we actually park our array at one cross-section. We just watch the motion of the small animal. Because we image so fast, we acquire data so fast, and so we don't have to worry about motion artifact at all. So we can see the motion, and you can see the motion quite well right here. This one centers around the liver portal vein region. This one centers around the chest region. So in fact, you can see the torsion of the heart and the heartbeat right here. And in fact, you can see some of the structures surrounding the lung as well on both sides. And the sternum is right here, and that's shown right here. This one centers at the liver lung region. Right, the liver region is right here. And you can see the detailed vessels. We don't have to inject any contrast agents. And so at the current wavelength, laser wavelength, we're targeting mainly blood absorption. So we're looking at mainly blood vessels using endogenous contrast. And we can also apply this for functional connectivity. I know this is a neuro-related audience, and so we can provide neuroimaging. This is endogenous contrast, functional contrast due to hemodynamic information. And so even in resting state, there's connectivity between two sides of a rat brain. So this is a whole brain imaging. And so this cross means a seed point. We look at correlation of the resting state hemoglobin signals. And you can see the symmetry right here between the two hemispheres. And you can see quite deep into the brain. So that's basically the whole rat brain. Now, of course, you can also do it for rat brain, for a mouse brain. And so rodent brains can be imaged quite comfortably using our technique and actually at a very high speed. Now, I know this is an EE audience as well, so please allow me to go through some of the details. So to build a high quality imaging system, you've got to pay attention to every step along the signal chain. And so starting from the view, and this slide demonstrates partial view versus full view. I start at a severely limited view. As I increase the view angle, the image quality improves step by step. And you can see when you have a full view, you get the best image quality. So whenever possible, a full view is always preferred, but sometimes it's limited by anatomy. So it's not possible to have a full view. Then you do your best. And there are other techniques, in fact, people are working on, so how we can overcome the limited view problem. And the other aspect is likely sampling. So I start with a very low sampling count, and then I start from 32, then I double each time, and you can see the image quality improve as well. The third aspect is image processing. It's important to process the image to highlight the important features, but we have to be careful because we don't want to over-process. And so oftentimes we want to compare the processed image with the original image, just to make sure we're not creating artifacts. The other aspect is the imaging frame rate. This is obviously very important. Sometimes, rats have a much higher heartbeat, as we know, than humans. And so they actually also breathe faster. And so you got to make sure your speed is high enough to take care of the motion artifact. So you can avoid motion artifact. And so this is at 50 hertz. You don't have to worry about any of those. But you can see if you have a lower speed, you may run into problems, especially the region where the breathing motion causes a lot of a large scale of motion. And so you can see here, if you sample at a lower rate, and all this motion can blur your image. So all those factors combined allowed us to get a good image. I don't have time to talk about the detailed image reconstruction. In fact, we use dual speed image reconstruction because the coupling medium has a different speed of sound than the biological tissue. And so if you account for that speed difference, the image quality improves. So excited by the small animal data, we decided to scale up for human imaging right away. And this is the breast imaging system. We have a laptop with an aperture of 22 centimeters in diameter. And you can see this little compression plate right here. So we only slightly deform the breast. And this is painless compression, very different from X-ray mammography. We just want to stabilize the breast and minimize the thickness. So we minimize the requirement for light penetration. And that's always a welcome compression just to improve the signal-to-noise ratio. And so this black ring is ultrasonic transducer. Again, we have 512 ultrasonic transducers that allows us to get single shot 2D cross-sectional image. And we can scan for 3D imaging. This system operates around this point. It gets multiple centimeters penetration, gets hundreds of microns resolution. And so this chart shows imaging depth versus spatial resolution. And so there are many different implementations, but there's a trade-off. But the depth to actual resolution ratio is roughly 200. And so you always have an adequate number of pixels in the depth direction to get a high resolution image. But you do have to run into this trade-off, and you don't really have the deepest penetration and highest spatial resolution at the same time. The physics doesn't allow that due to mainly light attenuation and ultrasound attenuation, which is frequency dependent or wavelength dependent. The wavelength determines your spatial resolution. And this is one of the first images we acquired from a healthy human subject. We use color to represent imaging depth. This is the nipple region. With a single laser shot, we get a 2D image. The data acquisition was finished within 150 microseconds, so you don't have to worry about motion artifact. Within a single breath hold, either 10 seconds or 15 seconds, we acquire a 3D image. And so we avoid breathing-induced motion artifact. The smallest vessels we can detect is about 1/4 of a millimeter in diameter. This is 4 times finer than contrast-enhanced MRI, which requires injection of gadolinium, a heavy metal-based contrast agent, and which is not entirely innocuous, as we know. And so we actually provide endogenous contrast imaging without injection of any exogenous contrast agent. So this is entirely safe. And the deepest penetration is roughly 4 centimeters. And you can see different cross sections, and that gives you an idea of the image quality we can get. So our technology has been commercialized by 2 companies. They have a human imaging version and a small animal imaging version. And this is one of the images they acquired from a cancerous patient. So later on, we implemented the first 3D photoacoustic microscopy, right? So you can see this microscope. This is a photograph. Let me just concentrate on this key component right here that's shown right here. And we generate a hollow cone beam. And that cone beam is refocused into the tissue. On the tissue surface, we have a donut-shaped beam. And so the core of the beam is dark. And we made the beam dark intentionally at the core just to minimize the surface interference. With a single laser shot, we get a 1D depth resolved image along the z-axis right here. And so that's based on the acoustic time of arrival, sort of like a sonar or radar, except there you deal with 2-way propagation of the signal. Here we deal with only 1-way propagation of the acoustic signal, and we can ignore the propagation time of the optical signal. And so single shot gives you 1D, then you raster scan this head in this water tray to get a 3D image. And there's a membrane window that allows us to couple light and sound. The object is placed below the membrane window. Now, of course, this is the initial version, and you can imagine you can implement this in many other variants. And so you can put this on a pneumatic arm. You can even miniaturize this for endoscopic imaging as well. And so to optimize the signal to noise ratio, we make sure the ultrasonic focus is confocal with your light illumination. And so that gives you the best signal. So the system, we just moved from here. You can slide down for higher resolution, but you do have to give up penetration depth. So this is millimeter scale penetration that gives you tens of microns, eventually even single-digit micron, even sub-micron scale resolution. So there's a huge range of scalability. So for this particular implementation, we can fire the laser at a very high rep rate, and this is close to a megahertz rep rate. And within a second, we can get a 3D image. And in fact, we can acquire information about oxygen saturation. This is a functional parameter, as we know, and we color code oxygen saturation. You can see the entire 3D cortex of a small animal. And this system is fast enough to see real-time neural response. This is again hemoglobin response to pulse stimulation. And we stimulate one side of the pulse, then you can see the contralateral side of the brain activate. Now, we want to see, as we know, both MRI is the workhorse for brain imaging, but both detects deoxyhemoglobin. So due to the paramagnetic property of deoxyhemoglobin, in fact, both does not see anything from oxyhemoglobin because they basically don't have any special magnetic properties for MRI to detect. And so we want to figure out exactly what we can do, how fast we can detect in terms of neural response. And we're actually doing a single impulse stimulation. This is an extremely short impulse to the small animal. We want to look at the brain response, and we'll dive down to the smallest vessel and see what we can see. Now you can see this propagation wave. The top one is due to oxygen saturation. The bottom one is due to the total concentration of hemoglobin. And so we actually have the advantage because we can choose the laser wavelength. We can see both oxy and deoxy hemoglobin molecules in parallel, and allows us to quantify two independent functional parameters, oxygen saturation and total hemoglobin concentration. And so this wave shows beautifully in small vessels, you can see features that represent the change of the neural response. And so let me just look at the details, because sometimes the movie only gives you the macroscopic feeling, but it doesn't tell you the exact detail. And so we start with a really short stimulation. And so we call it a single impulse, 40 milliseconds. We don't repeat it, it's just one shot. And then we look at response. And so the blue curve is oxygen saturation, and this top one is the total concentration of hemoglobin. You can see within about 130 milliseconds, and we see enough contrast to noise ratio. This is a 3 sigma point, and that allows us to call this a response. And so that's due to the oxygen saturation, and which is much faster response than the total concentration of hemoglobin, and which has a much greater time delay. Where with MRI, they cannot detect at such high spatial resolution. Of course, they're talking about human scale imaging. And so typically the neural response is multiple seconds in terms of the initial point where you can call that a response, 6 seconds, for example. And so we can potentially use this technique to help understand exactly what's being detected in MRI. And we can potentially also implement our technology for human scale imaging down the road. So another direction we took is to try to make our device cheaper. And so if you use an array of ultrasound elements, 512 elements, the system can get pretty bulky and expensive. And so can we design something that can make this much cheaper? And so this is using the concept of ergodicity. This is an acoustic ergodicity. And so this prism actually functions as an ergodic cavity. And what we do is we calibrate point by point, and later on, we can use a wide field to get all the signals in parallel. Then we solve the equation, and we reconstruct based on our calibration data, and we can figure out the point-by-point response from the entire field. And so in the end, you can get the image really fast. And so once this is implemented, you can imagine this can be miniaturized, maybe even for some portable applications. Can we couple this with a mobile phone eventually? And so that's an open question for us to answer down the road. And we can even push the resolution further to super resolution, down to sub-micron level. And this is a demonstration. With a very high numerical aperture, you can focus your light really tightly. You can get 234 nanometer resolution. This is diffraction limited, essentially lambda over 2. And now, if I use nonlinear photoacoustics, we can push the resolution to 90 nanometers. And we're looking at mitochondria. This is a comparison between the two versions, and that's an EM micrograph. And you can see we're revealing some of the internal structures within mitochondria. Now, there are a number of applications we can target. Human imaging, of course, is a very important direction. We're imaging human breast cancer. This is one example. This is X-ray mammogram of a breast cancer patient. And in fact, X-ray missed this tumor. And ultrasound picked up the tumor, and we placed a metal target on the breast surface, and we took another mammogram. And so this target this dot is not a tumor, but instead of the label, the metal label. And using our device, first of all, that's the nipple region. And we can see a dense collection of small vessels. In fact, you can work on the vessel density, and you can see this very high density population around the tumor. And we can also get a second contrast using the same modality, and we can quantify the stiffness because we can image so fast. We can look at the natural motion of the breast and figure out the stiffness. And the tumor region turned out to be stiffer than the surrounding tissue. And so the dual contrasts are always good to have because that can improve your detection sensitivity and specificity. And this one shows that because of the high imaging speed, we can look at the heartbeat-induced change in blood signals. And so we look at the pixel value right here, because as we know, the blood coming out of the heart goes to arteries first, then they get filtered, essentially filtered by tiny capillaries. So a lot of the mechanical motion will be filtered out. And by the time the blood flows to the venous region, and the veins don't really move as much. And so, when you palpate the heartbeat, you palpate, usually you palpate an artery. And so here we're essentially doing remote palpation, right, of the heartbeat-induced motion. And you can see this red indicates strong motion, and this region, blue region, is indicating a much weaker, much smaller amplitude motion. And so this red represents artery, and blue represents different veins. And you can see their magnitude is very, very different in terms of motion. And so this potentially can find many applications in biomedicine. Now there are extremity imaging, for skin level blood vessel imaging, and we can get really nice images from different regions, different parts of the extremities. And so very natural. And again, you can find many different applications. Diabetic foot is something we've been thinking about. It goes down all the way to revascularization surgery for monitoring. Just check out the viability of your skin graft on the spot, even in the OR, monitor perfusion. Now you could probably think of more applications. So we can do functional imaging. We want to push this to the ultimate limit, a single cell limit. And this is the system working at 1 hertz. And this is working at 20 hertz. They were for demonstration, but in reality, we operate at 200 hertz, and that allows us to get real-time images of red blood cells. And you can watch how they bifurcate. And in fact, we can do human-level imaging. So this is, we're looking at our own cuticle capillaries, and we can use different laser wavelengths to quantify the oxygen saturation. As a cell releases oxygen, it'll change its color. Of course, the color represents the oxygen saturation. And so this is a cell that's singled out of this ensemble, right? And so we provide a tool for physiologists to study how oxygen is released at most fundamental level, a single red blood cell level. Now we can also do a lot even ex vivo. And this is a recent work where we provide high throughput, label-free single cell imaging of oxygen consumption rates. And the standard tool is called Seahorse, and that allows us to quantify a group of cells. So there's no individual cell distribution, and because there's no sensitivity, and there's you just have to image a whole group in order to get enough signal. But we can actually sense at a single cell level. And we built micro wells to accommodate cells. One well accommodates a single cell, but we have thousands of micro wells. So we can sense all those cells and get a distribution. And this is, once you fire the laser pulse, you sense the photoacoustic signal that allows us to assess the current oxygen saturation because we have hemoglobin surrounding the cells. This is one example. There are histograms of normal cells and human breast cancer cells. And so this is a single cell level oxygen consumption rate plot. And you can see very clearly normal cells have a narrower distribution and on average has a lower oxygen consumption rate. And so this type of capability would allow oncologists to maybe follow a patient chemotherapy, this as a start. So there are many other applications as well. As long as you want to use seahorse, you may want to think about using this technology that gives you the whole distribution of the different cells. We know cells are highly heterogeneous, especially within cancer. And we also thought about, can we image? So far, I've mainly talked about hemoglobin as the main contrast, but can we image other type of chromophores in the body, other types of absorbers in the body? Melanin is another one, and that's strongly absorbing, and we can potentially use that to see without injecting contrast agents. And so the first idea that came to mind is melanoma circulating tumor cells. And we want to detect at a single cell level in vivo in humans. And so eventually we did that. And you can see a single cell swimming across the field of view, and we can capture that multiple times. Here we actually see a doublet, that two cells seem to be bound together and they travel across the field of view. And so there's no other technique that allows you to do label-free imaging of circulating tumor cells. And so in fact, when we image those set of patients, and the physicians basically gave us the patients, and there's no idea. They had no idea if this patient has any circulating tumor cells. And so we just tried it on the patients and we had to capture them. And so this can provide maybe earlier detection capability, follow up on some chemotherapy and see how the patient is progressing. And so there are many other possible applications you might be able to think of. As we know that the primary cancer doesn't necessarily kill the patient, right? It's really the metastasis. And the main channel of metastasis is through circulating tumor cells. And so now we have a mechanism to monitor even at a single cell level all these circulating tumor cells. Histology is the gold standard for disease diagnosis, but it requires tissue excision and requires time-consuming labeling and slicing, processing. And so this is why in the US, still roughly 1/3 of the breast cancer lumpectomy patients have to come back for a second surgery. And because the first time we did not clear all the margin. And so the hurdle, the main barrier is we don't have a fast enough diagnostic tool. And so if we can, within minutes, if we can diagnose whether the margin is clear for cancer, and then we can basically get rid of the second surgery. And so we want to develop a label-free version of histology. And by imaging the DNA and RNA within cell nuclei, we can provide image like this. And so we can tell the normal tissue apart from the breast cancer tissue by looking at the standard pathological features. And you can compare with the standard H-stain histology. You can see how well they correlate. In fact, if you pseudo-color our images, they look almost exactly like the H-stain images. And so our pathologists can already make diagnosis out of our images. Now, if it's ex vivo, you can also sometimes slice. This is an example of a brain, a mouse brain, and we can see DNA contrast, and we can see hemoglobin contrast, and we can see lipid contrast, and we can see a combination of different contrasts. And so even axons will show up in our images really nicely. And this is, again, without labeling. Now, if we tune our laser wavelength, we can get additional contrast. if we were to introduce any other standard optical contrast mechanisms, we can get yet additional contrast again. And so you can think this is as a very rich method for all sorts of contrast imaging purposes. Now, mid-IR is always highly desirable for imaging because it quantifies a different part of the spectral region that's related to molecular vibration. And so that gives you molecular specificity due to the vibrational structures of different molecules. But standard mid-IR imaging has a problem because the wavelength is very long. So first of all, the spatial resolution is very poor. And second, the absorption is so strong, there's no signal coming back in the reflection mode. And so the previous standard techniques work in transmission mode. That means you have to cut tissue before you can image. The third problem is water background. And so we actually invented this technique called ultraviolet-localized mid-IR photoacoustic microscopy that solves all three problems. And so the idea is actually pretty straightforward. And we start with the UV pulse. And if you don't like UV for some reason, if it's ex vivo, it doesn't really matter. But sometimes you want to say, I want to avoid UV, and then you can probably switch to another short wavelength light. And so the first UV produces a PA pulse, photoacoustic pulse, and we detect that signal. And then we introduce a mid-IR pulse to change the state of the material and the tissue. And of course, you get a PA pulse for free, even though this is not a critical one. And what's critical is the second UV pulse, and that will produce a second PA signal. And so what's important is to examine the relative change of the PA signal. And that's due to the intervening mid-IR pulse. And so this signal reveals mid-IR contrast, and that's what you're after. However, because the UV pulse can be focused much more tightly, and so you have a much finer spatial resolution. And the PA signal goes in all directions, so you can work in reflection mode. And also, this part eliminates a lot of the surrounding water absorption. So it eliminates, for the most part, the water background issue. And so this is a comparison of images acquired using mid-IR in the top row, and the bottom row are acquired using our technique, new technique. And you can see much finer spatial structures using the new technique. You can see cell nucleus, for example, and some tubules. Now, so far, it's all endogenous contrast. So what's in the body, and that's kind of nice because you don't have to stain, you don't have to inject any contrast agents. But with the injection of contrast agents, you can get additional contrast if the contrast agent injection is allowed. And this is an example where using endogenous contrast, we get tumor hypoxia using the hemoglobin contrast. But if we were to introduce some contrast agent, this is actually IR dye 800 conjugated with cyclopeptide to target integrin. So this is alpha V beta 3 integrin, and which is a hallmark of angiogenesis. And you can see this angiogenesis right here co-located with the tumor region. And so this is a form of molecular imaging using photoacoustics. So this is another example. So again, this is similar to functional MRI, right? And so here we get an image that's similar to PET. And so we use an optical glucose analog and to quantify glucose uptake. And you can see one side of the brain shows a little difference due to pulse stimulation. And so the glucose metabolism reflects the pulse stimulation. And you can see this photoacoustic imaging by itself is multi-scale imaging, multi-contrast imaging, but also integrates very well with standard imaging, including ultrasound, MRI, fMRI, and PET imaging, or maybe even SPIE imaging, because we can share the same type of mechanisms for different contrast. And so we can integrate all sorts of imaging modalities and to hopefully understand a given biological problem from all possible angles in an omniscale fashion. And this is a recent work where we use photoacoustic tomography to track micromotors. These are micro robots essentially. It's a capsule that can be used to deliver drug at a given spot and provide therapy. And so we can watch how the microrobots propagate in the GI tract. And when the microrobots reach the targeted region, and we can actually heat up the microrobot to release drug, and then we can monitor the response of the lesion. So let me talk about the second topic quickly. This is what we call compressed ultrafast photography. And this first generation works at 100 billion frames per second. Now, your standard camera may work at kilohertz frame rate, but we're talking about 100 gigahertz to begin with. So at this kind of frame rate, we can capture the fastest phenomena, namely light propagation, with a single shot. This is not a pump probe technique, right? Pump probe requires repetition of the event, and then each time you capture one snapshot, and then you change the delay, then you can resynthesize and form a movie. But here with a single laser shot, we capture the entire sequence. And you can see light propagation, reflection, refraction, and the two light pulses are having a race here. And this is a single shot fluorescence lifetime. Some of you might have heard of FLIM, fluorescence lifetime imaging or microscopy. This is essentially a single shot fluorescence lifetime. And the green pulse, we made our camera two-colored. The green pulse represents the excitation. The red actually represents fluorescence decay. Because the videos been slowed down 10 billion times, that's why you can watch the entire event. And so these are probably the slowest slow motion movies you're watching. Now, how do we build this device? Let me start from the standard street camera. So you start from a slit. The normal operation requires a very narrow slit, but we actually fully open. If you have a very narrow slit, then your white dimension information is lost. But we want to capture the white dimension. And so we have this input optics. We map the light onto this photocathode, which converts photons into electrons. The electrons are pulled by this accelerating mesh. So there's a voltage that pulls the electrons forward. And when the electrons pass through this pair of electrodes, we call them sweeping or sweep electrodes, we have a voltage ramp. So depending on the time of arrival of the electrons through this sweep electrodes, and the electrons will experience different voltages. And so they will be deflected to different vertical angles. And so this microchannel plate will amplify the electrons, and eventually we'll convert the electrons back into photons, so we capture them optically using a standard CCD or CMOS camera. And so that's how a standard street camera works. And so this sweeping part is really the key, because it allows us to freeze time, because none of the electronics is fast enough to respond at light speed. But if you can freeze time, you convert time into space. Then later on, you can take your time using a standard camera, and you capture the spatial information, and the spatial information can be converted into time. That's how you can get a really high frame rate. But a standard street camera only allows you to image a 1D object at a time. So to get a 2D, you got to repeat the event and scan along the other dimensions. So what we do differently is to add a coding mechanism, and we use compressed sensing concept. Let's start from the object. So you have this intensity distribution I versus X, Y, and time T, and we route it optically toward this digital micromirror device, which has a pseudo-random code. So these are zeros and ones. 1 represents reflection, 0 represents no reflection. And so we code the intensity using this pattern C of X, Y. Then we route this coded pattern through this route, through this path to this street camera. Again, this slit is widely open. So now the white dimension information actually is accepted. Now you go through the sweeping, this is called a shearing action, and that can be represented by function S of T. Then in the end, you integrate within your exposure time. So mathematically, we can write everything nicely into this single equation. And you have I, and that goes through 3 operators, coding C, shearing S, and time integration T. And then in the end, you get something equivalent to energy matrix E versus X, Y. So we have to solve this. Now, if you look at this equation, you may have spotted a problem. So on the left side, you got 2 dimensions. On the right side, you have 3 dimensions. And so I is unknown. E is what you measure. And so you have more unknowns than your number of measurements. And that's a problem, obviously. And so this is where we use compressed sensing, because in reality, your movie is highly compressible. The actual information content is far lower than just the raw representation of I. And so using the compressed sensing concept, we can solve this equation and invert it per eye, which gives us a movie. And so in the end, you get a movie of some really fast event. So coming back to this problem, this supersonic monochrome. So now for the first time, we can actually capture this event in real time. This is a simulated monochrome. So we have a light source propagating very fast, and we have medium surrounding this light source that slows down light propagation, that creates a superluminal situation. You can see this optical boom essentially. And so this is a monochrome observed in real time with a single shot of light pulse without repetition. And then we sped up our system further to 10 trillion frames per second. And so you can have a comparison. This is done in the original version. This is actually slowed down given all the experimental limitation, given the field of view and everything. And so this is working at 2.3 terahertz. And you can see the length scale is different by order of magnitude. And also we get more frames here within the field of view. So it's a much smoother movie. And this is a piece of glass. You can see light can scatter here, because we have some fog to scatter light toward our camera. And once the light enters the glass, there's no scatter light, so you don't really see it. But it'll come out the other side with all of that, as expected. So you can find a number of applications. Now you have a really fast light source, right? Standard LiDAR or sonar requires some sort of scanning, but now we have both XY information with a single shot. So you can do single shot LiDAR imaging. Now this is a lab demonstration. You have our CUP camera right here. If you have a flying object behind some scatter medium, that's mimicking cloud, for example, now you can see this moving object, right, really fast, because this is limited by how fast you repeat your laser pulse, and of course your frame rate of the data acquisition. More recently, we introduced the phase contrast into our technology. Right? The standard imaging, including our vision, right, is based on intensity contrast. But if we introduce phase contrast, now we can see object that's not visible using intensity contrast, such as a wavefront. And so this is a shock wave propagating in a transparent medium, and we can see the wavefront very nicely. And so you can use the same technique for a lot of cell level imaging. And because oftentimes without staining, the cell looks very transparent. It has a lot of phase contrast, but not necessarily intensity contrast. Recently, we increased the speed even further to 70 terahertz. I noticed that I'm probably running out of time, but so just to give you the key idea. So we start with a femtosecond laser pulse, but in order to get a very long image sequence, many image frames, we actually convert that single pulse into tiny subpulses through this essentially a Faber Pro device. It's called a beam splitter pair. Then we use a glass rod to disperse each subpulse to generate a color distribution. And so the color distribution will represents very fine time within each subpulse. And so you got T, that's actually the time separation between subpulses, but within each subpulse, you got finer spatial resolution. And so this is the key component that's shown right here. And so when we detect, we split different colors into different locations, and each color represents a time. And that's why you push the temporal resolution to the next level through this color coding, essentially. Of course, you have to solve the compressed sensing problem. And just to give you some idea about this improvement, and now we're looking at a curved medium. It's a light pulse propagating in a material on a very small length scale. So this is like 100 microns. And you can see the focusing of the light that generates curve effect. And this is also a phase contrast effect. And just to compare with the standard pump probe and also our previous version, you can see this is pump probe. It fluctuates a lot, and we have single shot. It's very stable. And so this is a detailed comparison. If you just look at the signal at this cross, and you plot them out, you can see ours is very smooth, and the pump probe fluctuates a lot. And our previous version has worse temporal resolution, so it doesn't really resolve the detail as much. All right, let me just finish with one last slide on the technique. And so, as we know, chaos is a frontier research area, but it's mainly limited to theories because by definition, chaos cannot be repeated. So how do you experiment and figure out what courses the event will take in a given realization. And so here, what we can do is, because we provide light speed imaging, and this is a cavity, it's a chaotic cavity, and this almost looks like a ping pong ball, but it's actually a single light pulse. And we're looking at a single light pulse in real time. And even if you change nothing, you just have 2 takes of the same event, and you will see actually the light pulses travel a different trajectory. And this is an overlay of the 2 movies. If we start from the beginning, you can see to begin with, they travel together, but if you wait long enough, they will split. And so because this cavity is not stable. And this is another one. We can even add a control within the cavity. This is a gate. It's a curved gate that's operating at light speed. And you can see we can open up the gate, let the pulse travel to the other side, because this rectangular pulse is actually a stable, non-chaotic cavity. But the minute you let in, open the gate, and now you have a chaotic cavity. So you can actually start by comparing the chaotic and non-chaotic behavior within the same device. And so this is where the technique is, right? So this is a plot of sequence depth, names the number of image frames per movie, and imaging speed. And so there are many versions of different technologies, but this is where CUP stands. So right now, this is the fastest and also the deepest in terms of sequence depth. And so this is a very active research area right now. Finally, if you're interested in more information, please visit our website. There are a number of papers posted there as well, and a couple of books for more details. One on photoacoustics, one on biometric optics in general. And I'm very grateful for Caltech for building me a dream lab. And so this is one of the floors of our lab. And we have nanoscopic imaging at the proximal end. We have human scale imaging at the distal end. And so this is a multi-scale or omniscale imaging lab to begin with, using photoacoustics and some related techniques. And credit goes to my lab members. And we do have openings for graduate students and postdocs as well. Please let me know if you're interested. And we're mainly funded by NIH, NSF, and NASA. Thank you very much.
Thank you very much, Dr. Wang, for the fantastic presentation. Are there any questions from the audience? You can raise your hands and I can unmute you so you can speak. So I think as the audience are preparing their questions, maybe I can ask one for our students in neuroengineering, right? What does it take to be able to directly imaging neural activity using photoacoustic imaging? And what do you see as the opportunities and challenges there?
It's a great question. So, we're extremely interested in that direction. In fact, we received several Bren initiative grants to push that direction. The very immediate application is endogenous imaging. As I demonstrated, we can image the resting state functional connectivity using hemoglobin signals. And so, in fact, we're pushing the human scale imaging. We have some results, but it's not published yet. So that's a extremely exciting direction for us. It's highly complementary to functional MRI. There are challenges for human scale because the scope is in the way, but ex vivo imaging has demonstrated we can actually provide penetration through the skull, both optically and ultrasonically. And so you have to use the right laser wavelength, of course, and the ultrasonic propagation requires a lower frequency. So typically for ultrasound imaging, for example, for breast imaging, we may actually use multiple megahertz, but it turned out for transcranial imaging for adult human skull, we have to lower that down to maybe less than 1 megahertz. And so we found that 0.8 may be an optimal number, and that still allows us to get about maybe a millimeter or 2 resolution, which is adequate, somewhat comfortable to bold MR resolution anyway. Yeah, but we could provide faster imaging and potentially much lower cost. We compare with the 7 Tesla system, which is $5 or $6 million, extremely clunky and very hard to maintain, and it creates noise and has incompatibility. If the subject has some implants, then you really cannot use MRI. And so we can bypass a lot of the limitations using our device, but that's still an area that's being researched. And so we need to first overcome the signal-to-noise ratio and problem. And the other one is a de-aberration, because the acoustic wave comes out of the skull with a distortion, right? Because the skull is sort of like an bad lens, right? So it's going to generate distortion, and we have to de-aberrate somehow using a really sophisticated inverse problem. And in terms of small animal imaging, in addition to functional contrast based on endogenous hemoglobin, and we can potentially look at blood flow velocity, right? So very similar to ultrasound Doppler, and there's the photoacoustic Doppler, in fact, that we can potentially leverage. There's also a lot of interest in terms of labeled imaging. So the GCAM based for calcium level imaging, or maybe even more exciting is the voltage type of imaging using either proteins or dyes. We're open to any one of them. So it is possible. So our field has demonstrated, including my own lab, using GCAM, we can get photoacoustic signals as well. But GCAM works at the wrong wavelength because it's a green wavelength. And so that means the light penetration will not be very high. And so will not provide deep brain imaging, whole brain imaging. And so we're waiting for a near infrared version of GCAM or voltage or / protein labels for voltage sensor. These are, as you know, generally called the labels for either calcium, surrogate, or voltage. And so the hardware is ready, as I showed for whole rat brain imaging. We can actually do it with a single shot. So within tens of microseconds, you get a 2D image already. So there's no competition in terms of whole brain imaging, whole mouse brain imaging. In terms of voltage contrast, as soon as we have the right wavelength for voltage labeling, we can potentially demonstrate that right away. As we know, for voltage labeling, optical contrast is really the only availability. So there's no MR counterpart, no CT counterpart, no ultrasound counterpart. So optical imaging, standard optical imaging like dual photon or confocal or any other modalities, they will not give you the penetration required to see the whole mouse brain or whole rat brain. And we can see either rat or mouse brain, right, in its entirety.
Yeah, thank you so much.
Sure.
There is a question from Mary Jean. You can talk, Mary.
Hi, Dr. Wang. So my question is for the photoacoustic microscopy, do you envision to see it applied to in vivo imaging in addition to the current ex vivo on fixed tissue? Thank you.
Right. It's actually done in vivo already. And so you can do it in vivo. There's no limitation on either ex vivo or in vivo. So we can use it to image mouse brain, human skin, for example. Also possibly intraoperatively. So you can build a device, potentially handheld. I don't have time to talk about this here. We had a study that was transvaginal imaging for preterm birth. And so you can implement it endoscopically as well. We can miniaturize our probe so small that you can send it to the GI tract, for example, right? So very much like, the standard endoscopic imaging, except we introduce our technique, not just either optical, as you know, the standard endoscopy is purely optical, and ultrasound has a ultrasonic counterpart for endoscopic imaging, but we have photoacoustic endoscopic imaging as well. And in fact, the field is even pushing intravascular imaging. You might have heard of IVUS, intravascular ultrasound imaging, but there's also intravascular photoacoustic microscopy, and that's used for plaque detection, because we want to detect the vulnerability of plaques. So yes, it's possible to do in vivo photoacoustic microscopy.
Thank you so much.
Sure.
If not, let's thank Dr. Wang for his presentation. Oh, hang on a minute. We have one more question. Can we answer that?
Yeah, sure.
Yeah, Brian.
Can you all hear me?
Yeah, yeah.
Oh, yes. Great presentation, Dr. Wang. It was really interesting. It's just, I guess, has there any been studies or, I guess, the effects of, or reading the effects from stroke imaging by chance? Use method of recording imaging?
Yeah, that's, so you want to image the brain stroke. So that, we've been talking about that. So for now, we're talking about functional imaging because that requires less penetration. The stroke can happen sometimes quite deep in the brain, and so it's a more challenging application. As a starting point, we say, well, let's focus on the cortical region of the human brain. But we are developing a microwave version of photoacoustics because microwave can penetrate much deeper than light in soft tissue. And so, that actually has maybe, greater immediacy in terms of stroke imaging. And because there's microwave contrast in stroke as well. That's a great question.
Thank you.
Thank you.
Yeah, thank you all.
Thank you, Dr. Wang.
All right.
Thanks for.
Thank you.
Yeah. Bye.
All right. Bye now.
Bye, everyone.